I'm very delighted to introduce Professor Hugh Watkins. Hugh is based in Oxford, and I'm sure he's well known to the audience here. He's going to be talking to us about the role of genetic testing in borderline HCM phenotypes. Thank you, Hugh.
Great. Thank you very much, Nora. Let me get my slides up. Terrific. Yeah, so I think it is timely to revisit this old chestnut. We've got a little bit of clarity that we didn't have before that I think does shift the goalposts a little bit. So as you just heard, and as is well known, the framework we have to work in hypertrophic cardiomyopathy is the Mendelian framework, because it's the only bit we've really known with certainty. So we do our genetic testing on the presumption of Mendelian disease, but we know that that's not the case in actually more than half of the cases that we see, because they will be gene panel negative. And even when we do have a variant in one of the myofilament proteins, we get this huge variability in individuals. So there's clearly other genetic or environmental factors. And there's been a big missing, unexplained bit of HCM. And the consequence of that is that all of our genetic testing just looks at that Mendelian model. And if you do a gene panel test and have a negative result, it's been considered that that's just a null result. It doesn't take you further forward. It doesn't exclude anything. And that would make it not useful. And that's what's driven the standard practice that we adhere to and that's in the guidelines, that we're really using our genetic testing in individuals who meet the diagnostic criteria. Because if you've got someone who doesn't, but maybe possibly has an abnormal heart, and you get a negative result, we would have believed it doesn't take you any further forward. I think that's shifting a bit now, and that would be good to look at the data, which are recent, and then come back to this question. And this is all about getting a better understanding of what is going on in the patients that don't have a myofilament mutation. And I used a shorthand sarcoma negative because I haven't got a better term. I'd love to hear a better suggestion, but this has some utility and currency. So sarcoma negative HCM, exactly as you just heard from William, is at least half and actually more. These are the results from the Oxford Genetic Testing Lab, and I think we are no doubt enriched for individuals that are thought likely to have familial genetic HCM. But even in our enriched population, about half come up with a negative gene panel test. It's about 2/3 or 3/4 in other series. And this has limited the diagnostic yield and therefore the utility of this kind of gene panel test. We've predicted for many years that what's going on in this half is not Mendelian genetics where we haven't found the gene. Instead, I've always assumed that it's either oligogenic, which is a few genes, or polygenic, a variety of common variants driving it. And the reason for that is that we see aggregation in families, but we don't see the extended families that you get with a single monogenic variant. So the goal for the last few years has been, how do we make the gene testing that we can do useful so that the negative result becomes useful? And there have been some pointers that we've been heading that way for a while. There's been quite good data from the large cohort studies that the gene negative individuals do better. We know certainly from our own experience that if you have a proband who's sarcoma gene negative, you're much less likely to find serious disease in the first and second degree relatives. And you might wonder whether that's sufficient to alter the way we manage those families. And we've always speculated that if we can identify what's going on here, we will find genetic variants that may well be the modifiers that drive the variable phenotype and penetrance where you do have a sarcoma mutation. So this has been an important, but until recently unmet goal. We do know quite a bit about the phenotypic characteristics of this group. And again, I come back to our HCMR study, and you've just seen this figure from the slide. And it was quite striking that when we look at large real world experience, many sites, many countries, both sides of the Atlantic, you do get these groups that can be dichotomized on the presence or absence of a rare sarcomatic variant. And it was unexpected to me that features like left ventricular outflow obstruction and apical HCM, are actually more characteristic of the sarcomere negative group. These would have been considered, hallmarks of HCM back in the 60s and 70s and 80s, but in fact, they're more characteristic of the group that don't have sarcomere mutations. So to take this forward and to test the polygenic common variant contribution, we have been conducting over the last year or two a very large-scale common variant genome-wide association analysis in the HCMR study. And I think it's given us quite a lot of clarity that we just published a couple of weeks ago. So we did an analysis of all of the cases in HCMR, 2,500, typing common variants throughout the genome, many millions of variants, using as controls the healthy population in UK Biobank. And what we see, and this is a technical but really important thing, really striking, this is the genetic contribution from these common variants. So 35% of your risk of developing HCM is driven by these common variants that we all have. We're excluding rare variants, we're excluding the environment, and we're left with 35%. That's a very much bigger number than I would ever have guessed. In another study on patients from Oxford that have phenotypes that we thought would yield a myofilament mutation, but didn't, they're young, they have affected family, they have the characteristic shape of the heart, we get amazingly high common variant contributions. You might have thought in this group there were rare variants, new genes that we were missing. That is not the explanation for sarcomere negative HCM. Instead, it's a fantastically powerfully determined genetic trait determined by common variants. So 67% of heritability. That is much, much higher than you would see with diabetes or coronary disease or other common diseases. A remarkably high number. So given these numbers, we were optimistic we would have power to find new genes, and we certainly do. And I can summarize those on a new slide. So these are genes or regions of the genome in which there are common variants. That these are the frequencies, so all of us will have some of these genes, which at a very high level of confidence contribute to your risk of developing an HCM phenotype. And if you look at the gene names, what you'll find is they do not include the genes in which rare mutations cause HCM. And that's predictable. That the mutations in myosin and troponin that cause HCM change the coding sequence and the structure, but the common variants that we all have change the amount, they change the expression. So it's a different bunch of genes. Some of them are known cardiomyopathy genes, and some of them have a role in DCM, but they were all bar one novel for HCM. And when there's a role in both HCM and DCM, then as you'd expect, the direction of effect is opposite. So the variant that makes you more likely to get HCM protects you from DCM and vice versa. So there's about a dozen of these on this slide. We're up to about 20 now because we've done more work on this that's not yet published. Each of these variants has a relatively modest odds ratio. So this one increases your risk of HCM by 28%, this one by 79%. So they're not big compared with the rare variant effects, though they are big for common variants. And you would predict that you could combine them all to aggregate and say in an individual, how likely are you to be protected or at risk of HCM? And we've done that, and it works quite well. So this is, again, a plot of odds ratio. So this is average risk, this is half, and this is double. And what we've plotted is the 20% of the population that have the lowest number of these common variants against the 20% that have the highest number. So if you look at this group here, imagine you've inherited a myofilament mutation, an MYH7 mutation, that's going to give you, let's say, a 50% lifetime risk of significant HCM. 20% of the people in that group have a bad luck hand of common variants that will double their chance. So if you start out at 50% and you double it, you're very likely to get disease. But another 20% will have a half chance, so they probably won't. And I think it's these common variants that are determining whether you'll get a phenotype, whether you get it early or late, or whether it's severe. The effects are bigger. There's a bigger common variant contribution in sarcoma negative HCM, but to get sarcoma negative HCM, clearly you're not going to get it in the top 20%. It's going to be in the top 1% or 0.5%, which is why we see the population prevalence that we see. And then you can get the disease purely through these common variants. So we've got a better understanding of two routes that can take you to have HCM. The one we know about for a long time is you have your rare penetrant mutation. And on top of that will be the impact of these common variants that we've now discovered. But you can get to an HCM phenotype without the rare variant, solely with the common variants. And I think that's what's going on in the large majority, probably not completely all, but a large majority of the 60 or 70% who come up negative on a gene panel test. It's a polygenic trait. One of the nice things you can do once you've done these genome-wide studies is you can actually test for the causal role of environmental factors if they themselves are genetically determined. This is through Mendelian randomization. I can't explain the details, but it will be well known to people who research in the area, and it's all in our paper. And it gave us some really quite punchy results. So we know, and this was seen in HCMR and in SHARE, that patients with HCM, but particularly sarcoma negative HCM, have higher blood pressure, higher BMI, higher waist hip, and more diabetes. That could be causal of their cardiomyopathy, or it could be secondary to inability to exercise or advice to not exercise. And when we test for causality, we find that type 2 diabetes does not have a causal role, but each of these other factors gives a significant odds ratio. This is your increased risk per standard deviation. So these are all contributing a causal effect on your risk of HCM. And the standout result, which I think is going to change practice straight away, is the role of diastolic blood pressure in the sarcoma negatives. So here, one standard deviation, which is 10 millimeters of mercury of blood pressure. So the difference between being 120 over 80 as opposed to 120 over 90 gives you a 4-fold increased risk of sarcoma negative HCM. So we really need to jump on blood pressure and treat to lower thresholds in families who have sarcoma negative HCM individuals than the rest of the population, and could probably prevent a lot of disease that way. So the bottom line is that these are surprisingly different disorders at the genetic architecture level. The sarcomeric HCM, we know about its rare variants. We now know about the modifiers. We know what many of them are. But the sarcoma negative HCM, which is more than half, is a polygenic trait. It's not missed genes or missed rare variants. And because it's polygenic, it's very unlikely to recur in distant family relatives. And I think the fact that we do clinical cascade screening the same way in families who don't have a gene test result is wrong and will have to change. And because of the comorbidities, we now know that blood pressure is centrally important. And in fact, in sarcoma negative HCM for many people is really just an exaggerated response to moderate hypertension in adult life. And it puts this disorder on the same spectrum as hypertensive heart disease and HFpEF, a shared genetic etiology. So to bring that back in my last slide to what does this mean for testing in borderline cases? I think in the context of what you think looks to be familial HCM, nothing has really changed. If you've got indications in an individual or a family that it looks like sarcoma positive HCM, definitely you still want to test the most clearly positive individual. If you're referred to someone who's borderline and somebody else over there has a clear phenotype, you need to get DNA from the other person. But if the best you can do is test a borderline person, maybe the severe proband has died and there's no DNA, I believe strongly that it is worthwhile because the yield is quite high and it will change the way you manage the family. But if you've got nobody who looks affected at all, I think the result is, as it always was, not profitable to test. The bit that I think is different is where you've got a confounding diagnosis. So if you've got an athletic 20 or 30-year-old, or a hypertensive 30, 40 or 50-year-old, or an obese 40 or 50-year-old who's got some features that could be HCM or could be secondary, we used to say, no point testing because a negative test rules out nothing. I think now a negative test rules out a lot. And you need to rule it out, because the thing you don't want to miss is sarcomeric HCM. And the very last case from William was a really good example of that. So I think it is now reasonable to test in this setting, because you can rule out something important. And if you get a negative result, you can say to your 50-year-old hypertensive man, actually, I'm less worried about you. I'm much less worried about your kids. I'm not going to even consider cascade screening into your further relatives. This is a very different disease, but tell me about your blood pressure. I don't know that things have changed in how we do testing in the group that just look to be something different. If you've got somebody who's got the angulated septum, they're older, they've been in hypertension for many years, probably your yield from testing is low. It might be safe just to assume you've got the polygenic HCM. Depends a bit on cost. And if cost comes down, it would be quite nice to test more. But the important thing is they're probably part of the same spectrum of disease. And you'll be managing this group by looking at comorbidities, particularly hypertension. And I think that will give you a different outcome. So whistle stop, I hope it was clear. Because it's research and recently published, keen to highlight Andrew Harper, who many will know. Kate has already been come up on other slides, and Liz Ormondroyd, and the statistical and analytical team in my group in Oxford. Many thanks.
Thank you, Hugh. That was really exciting and interesting data that you've produced recently, and shedding a lot of light on the polygenic nature of hypertrophy. And just following on from that, while we